Okay, let's briefly look at some applications of microbial metabolism. Microbes in food production, the byproducts that occur from fermentation by bacteria can be used to create various foods that contain either ethanol from ethanol fermentation or acidic compounds like lactic acid from lactic acid fermentation. So we'll look at three foods here: bread, soy sauce, and sauerkraut. So first, bread production depends on Saccharomyces cerevisiae, or what's also known as baker's or brewer's yeast. And I went over this in the fungi lecture, but here we're discussing how, right, these yeast, they can perform ethanol fermentation. So when they're deprived of oxygen, right, they'll resort to fermentation and, remember, stick the electrons back onto the pyruvate and generate ethanol as well as carbon dioxide as a byproduct of their metabolism. So those are the things that'll kind of come out of the yeast when they're mixed in with the bread dough. And of course, the carbon dioxide is going to provide the gas to make all the the bubbles that you see inside the bread, as well as allow the bread to rise, while the ethanol is going to get boiled away whenever you heat the dough within the oven. Okay, next, soy sauce. It's produced by, you crush up a bunch of soybeans and wheat and with like salt water, and then you add a fungus and various other microbes. So the whole process takes several months of fermentation, and the fungus of choice is usually either, these are two different species of mold known as either Aspergillus oryzae or Aspergillus sojae. And then, so these are actually going to be, they're going to be inoculated onto the mixture of grain and soybeans. So you're intentionally putting these guys on, whereas these other microbes like yeast species and lactobacillus species, they kind of just pop up naturally because they're already within the mixture. And these lactobacillus, you'll see these in the context of bacteria that perform lactic acid fermentation. Yeah. So initially, the Aspergillus mold, it's once you add it to the mixture, it's going to break down all the sugars and proteins using its hydrolytic enzymes, right? Enzymes that can perform hydrolysis, where they use water to break down complex macromolecules into smaller pieces, essentially. So after that occurs, then the yeast and bacteria that are there will come in. Now that the food has been broken down, right, they can go ahead and take the polysaccharides from that mixture and then perform fermentation, which releases lactic acid from the bacteria and then various alcohols from the yeast. And that's where the flavor of the soy sauce is going to develop. And then this mixture is later transferred into brine or high salt concentration water, which again gives it the saltiness as well as restricting the growth of microbes that you don't want, such as like pathogenic microbes that might grow if you didn't add that salt. And this mixture of soybean and grain is also called koji. Okay, next, sauerkraut. So sauerkraut, I'm sure you've heard of it, is fermented raw cabbage, and this one relies mostly on lactic acid bacteria for the production. So it's different from the two previous foods that we saw, because here we're not actually intentionally adding a microbe, like we're not adding yeast or we're not adding aspergillus, right? The microbes that will do the fermentation are already within the cabbage. So all you have to do is you just put them, you mix it with salt and water, and then you just place it inside a sealed container so that the oxygen is deprived, and then all the lactobacillus and leuconostoc species that are already within the cabbage will perform lactic acid fermentation. So leuconostoc mesenteroids, this is usually the first kind of bacterium that kind of predominates in the earlier stages of fermentation over the course of a month. And these are lactic acid bacteria that, of course, will produce lactic acid and then slowly lower the pH of the mixture. And actually, once the pH is lowered sufficiently, right, these guys will actually die off because it even gets a little bit too acidic for their survival. So then these two other species called Lactobacillus brevis and Lactobacillus plantarum will come in and start predominating after leuconostoc. And these, again, are lactic acid bacteria that can survive in higher acidic conditions, and they'll continue to do the fermentation process until around a month, and then you take out You open your container and then you have your sauerkraut. Okay, biofuel production. So we already talked about this in the microbes and agents list protist lecture. And there we saw that you can use photosynthetic algae, such as like Nanochloropsis species. You can grow them either in these bioreactors or in these big raceway ponds. And then you can keep feeding them, and then you can extract the high energy lipids that they naturally produce and purify those to use those as biological fuel sources. And of course, those are renewable sources because you can keep adding food and they'll continue to produce those lipids for you. And then we talked about some of the shortcomings as well as some of the advantages of this process. So I'd encourage you to go and review that lecture again. And then nitrogen fixation, I discussed this in the lecture I just did over the microbial metabolic strategy. So you can go and review that for nitrogen fixation. And these two are here because they're technically in the rules within the section with the rest of these other foods. But, I just wanted to mention where they're located in the other lectures in case you thought I was missing out these. Okay, that's it.